Welcome back, everyone. Hey, I was coming on today because I found this video with Dr. I think John Campbell. I've been following him ever since back when the P was really active and up until now. And at one time, I think, if I remember correctly, he was one of the ones that maybe possibly was pushing the you know what that's in the arm, that goes in the arm. But now I see that he is getting more knowledge of what what goes in the arm or when in the arm was all about. Y'all, I am really disturbed. I really am. I'm disturbed because I remember I did research over and over and over and over concerning this. And I was looked upon when I voiced the research that I had obtained. I was looked at as a person that was, what they call it when spreading lies or whatever. I've never been a person that even operate in lying. I'm not saying that I haven't never lied, but to operate in a lie, just to operate in something so serious, no, that would never be. I was doing the research to make sure that my family was protected. So in my research, I found things that made me turn a side eye to that which would go in the arm during the pandemic. So I kept my side eye and I started sharing the things that I had found, and to try to help people to just slow down, just stop, look, and just slow down for a minute to see what's really going on and what is really happening. First of all, I know that the things in this world, some have act as if if some of the people in this world just love you so much. And I just couldn't see that because I saw other things that the people in this world have allowed to happen. So I know that couldn't be true. That you want to do this protection so bad. So a lot of things was raising a red flag in my head, which I'm thankful that it did. But I remember this man. I don't forgot his name. I have his videos. But it's a funny name. But he said, you never give that which you put in the arm during a P, which, what was going on in 2020. You never give a V during a P. So he said that if you do, it would find a way of an escape, because the regular immune system is looking to fight off things, but you have put in the thing that goes in the arm. You have put that in. So now it has shut down your regular immune system. So it now has what it put into your body. It has that for protection now. So it's looking for that which you have made it to look for, which was the very first one. But now it has made a way of an escape. Just like in a football, when you're playing football, yeah, you going in and down the center, but then you have people that's guarding and protecting it, so therefore it make its way around it. And that's what, once it went to the second supposedly pandemic, the one that there was another one that came out. Well, in my mind, I'm still watching. And I'm saying to myself, that's what the man was saying, that it would go around. So it is now another one on the loose. So it's one after another that you're putting in your arm, and then it's finding a way of an escape. So it's creating more of the pandemic. So I looked at that and I'm like, I'm still side-eyed and I'm standing still. So my thing is, we were beat up about doing research. Who beat someone up about doing research? Who looked down upon somebody about doing research? Tell me that. But anyway, y'all, I found this video that talks about DNA, and I'm just gonna need you all to just open up your mind. And again, me trying to speak out and say things that maybe some would understand, some don't want to hear, and some would put me down for. But look at this video and really listen to what this gentleman and Dr. John Campbell is now saying. Lord, y'all, at this point, at that point, we really need God. We have to stay prayed up. Stay, listening to God's voice. If he's not telling you to move forward with something, please don't. But anyway, check this video out. And I pray blessings. I pray for the world.
Well, if I'd known that, I wouldn't have had hesitancy, or would have had abject terror and rejection, as I suspect the entire world's population would have done. It's just, we understand why people are angry.
It really is quite disastrous, and it's very saddening. And I have to use the expression in this country because it's just calling a spade a spade. Yeah. Our health authorities knew about this. They still deployed them, multiple doses, and so that's multiple deliveries of synthetic genetic cargo, and therefore they poisoned the DNA well, the natural DNA well of this country. Now, they're going to have the mendacity to come back and say, prove it. Right? They're going to be so arrogant and bold and desperate, they're going to say, prove it. Prove it that we've altered the DNA of the country. And everyone watching this has to, if they hear any politician or bureaucrat say that particular thing, prove it, say, mate, it was your job. You get paid the billions for your ministry every year to prove it's not going to affect us before you give it to us. Don't put the onus on me, now that you've injected me multiple times, to prove that something that you told me was safe and effective hasn't altered my genes. It was your job, always. And the legislation clearly says that globally, that the regulator has.
And it's clearly a genetically modified organism from what you've said. I hadn't realised that before. It's quite powerful, really.
And let's just look at that, just that issue of what types of genetic material are we speaking about here. With the standard product, we've got the LNP-mod RNA complex, right? The two go together. You haven't got a product. The mod RNA alone, if you just inject that, it gets destroyed in the bloodstream very quickly. It needs to be protected by the LMPs.
Just be recognised as foreign material and immediately eradicated.
Well, in science, they call it a complex. The two have to go together for the thing to work.
Yeah.
Now, we're talking about modified synthetic RNA here.
Yeah.
We've seen on websites, and they're still persisting with the lie in this country on the TGA's websites, that it doesn't enter the nucleus. Right? Well, that's wrong, because we provided the science papers to the Secretary of Health in this country. We did that in early 2022, showing studies that indeed this mod RNA does make its way into the nucleus of cells. When it produces the synthetic spike protein, the spike protein has a section in it that's called an NLS, a nuclear localization signal. Now, what that effectively is a portion of that protein, the spike protein. It's a key that gives it entry, grants it entry to go into the nucleus, to open the nuclear envelope. And there's this other aspect of the spike protein. It has an affinity with the actual mod RNA which creates it to passage it, give it a ride, in other words, to take the mod RNA into the nucleus as well. Now, the science papers are there showing this. We've got the Oldham paper, we've got the Quinn paper, we've got several, and they're in all the links that will be shared in the show notes later. So we've backed everything. And so we went to the TGA when we were suing the Secretary of Health, and we said, look, we can still fight this thing out in court, but stop putting this persistent lie on your website that it doesn't go into the nucleus. It does. We've given you the material. We did this by a private and direct note appealing to the Secretary of Health, stop the rubbish communications. Okay. Again, he had the arrogance not to respond. He didn't refute. He just didn't respond. Now, we were just doing the proper sciencey thing here. It's like, here's the information, and he doesn't do us pay us the respect to come back and say your science is wrong. He just stayed silent. We had him, okay, but he persists with this lie. Now, so let's just take it for granted, and can be found in all the links later on, that it does go into the nucleus. Mod RNA. So we're talking about RNA, DNA, different things. Exactly, sure, but there's this thing called reverse transcription. And that's what the Oldham paper shows with the Pfizer product, that it reverse transcribes into DNA. And there's a type of enzyme called reverse transcriptase, which is part of what's called line 1, which is found in all cells. It's found in more cells that are undergoing rapid cell division. So you'll find that around the ovaries, for instance, or a newborn, the embryo, because they're rapidly dividing. So there's lots of line 1 activity there. Therefore, there's lots of reverse transcriptase. Reverse transcriptase is a way of, to put it in pretty straightforward terms, read the RNA and say, okay, I can convert you into DNA. Now, just as DNA gets read and converted into RNA, which goes off and creates proteins, it's just the reversal, effectively, of the same process. But on this occasion, the enzyme reads the RNA first and translates it, transcribes it, and translates it into DNA, and slips it into the DNA. The science on this is called retroposition. Retroposition. Now, all these statements since 2021 that this stuff cannot interfere with DNA is based on absolutely no science, because they never performed those studies that we spoke about, John. But what all the health regulatory bodies have known for, I think it's going on nearly 50 years now, is the science of retroposition. That is, how RNA gets reverse transcribed into DNA. Indeed, there was a Nobel Prize awarded on the science in this area. It is so highly understood. But yet when the mod RNA drugs came out, they denied the science even existed.
I've known about it personally since the 80s from the
There you
AIDS
go.
pandemic.
There you go. Right. Well, yes, of course, because that's part of the mode of action there.
Yeah.
I wrote a paper which was subsequently peer-reviewed and published called, what is it, The Canaries in the Human DNA Mind, which speaks to the Australian and European GMO laws and makes it very clear that they apply, but also goes into the very well-established science on retropositions. And one particular paper which brought this to everyone's, or sought to bring this to everyone's attention in 2021, which was titled, "What About the Science of Retropositions?" This fellow was, this PhD was saying, this science has been around for 50 years, but we've got all the regulatory agencies saying that there's no chance of reverse transcription. And he was very politely saying, they're all lying to you. All of these regulatory agencies are lying to you. So we've got that. The main constituents, ingredients of Moderna and Pfizer is, receiving trillions of particles of this modified RNA, hundreds of billions, if not trillions. But it's in that range per.
Yeah, it's pretty sure it's trillions. Yeah.
Yeah. So it's, your chances then, after you've taken a second, a third, and fourth, boosters and all the rest of it, are just compounding as to respect to the probability of reverse transcription occurring, and therefore your DNA has been changed, and therefore something's going to occur with your DNA. It's either wherever it gets integrated,
Yeah.
the genes in that area are either going to be upregulated, in other words, they're going to express more of something that they shouldn't be expressing, or under-expressed, or silenced. Now, whenever you do that, you're completely blowing the rhythm, if you like, of the way that those genes should be normally functioning. And when you disturb the equilibrium, well, then you're heading down all sorts of paths to what we call generally genetic disease. And that's, just throw a dart at the board on that one.
And cancer's a genetic disease.
And then we ultimately end up with disturb the genome sufficiently, and the cell just says, let's just go cancer. That's what the cell does. Like, look, this is a mess. The DNA is not working. Let's just go to cancer mode. That's what cells sort of essentially do.
Yes, it's multiple mutations essentially. Yeah.
That's right. So we're in that territory, and we know, despite health authorities and governments paying off mainstream media to shut up about turbo cancers and the rise of turbo cancers. Look, Professor Dalgleish been on several times. God, how many credentials do you need until you listen to a man with that authority, with that expertise? How long can MPs say he doesn't know what he's talking about? It's absolutely despicable and disgusting, the contempt that this man who's alerting everyone as to what's going on is just being shuttered. Right? And he's not the only one. They're everywhere, these specialists around the globe who are saying it's real, the phenomenon's real. And it's not just the cancers, it's all the other spectrum of diseases and disorders that we've seen arising out of the adverse event reporting systems globally. Okay? They run into the thousands of different conditions. Thousands of different conditions. And you say, well, how can just some LNP and mod RNA create that variety of adverse events? It's like, well, buddy, this stuff isn't just going into the cytoplasm, it's going into the nucleus and disrupting the activity of DNA, the blueprint for life.
And all over the body as well.
That's right.
Systemic distribution.
This stuff won't be just integrating in just one specific site in our chromosomes for every person. It will be doing it in many, many different areas. And so if it's disrupting different segments, different sequences, a multitude of different sequences of DNA, well, then you're going to get a multitude and variety of genetic disorders or diseases, right? They won't be identified immediately as having a genetic disease origin, but they'll have a multitude of disease. When we start to underlearn the new pathways of where these diseases are originating from, with more discrete studies and analysis, the scientists in the lab are going to go, oh, hell, that disease is normally originates in, that part of our physiology, but this is tracking back to actually DNA dysfunction, right? So a lot of this stuff's going to in due course. That's what we anticipate. And I'm not just I'm not conjecturing here. A lot of our team work with a lot of PhDs and professors and associate professors of medicine, and we have these discussions. These are the people who know and who share their ideas with me so I can share them with you. So we've spoken about the mod RNA. That's one aspect of it. But correct me if I'm wrong, have you not spoken with Kevin McKernan in the past, the geneticists?
No, I haven't, no.
Oh, okay. I thought you had presented to your viewers the DNA contamination that's been discovered.
Oh, we've talked about DNA contamination, but not Kevin McKernan.
Okay, well, it was Kevin McKernan who was the one who raised the flag first.
Right,
what an appropriate individual to do it. I had been waiting.
Yeah, the DNA contamination in the simian virus.
That's right.
Yeah.
So Kevin came out with that in February of last year. Now, I knew of Kevin beforehand, and I'd been watching him, I guess, from the sidelines, so to say, throughout 2021 and 2022, wondering if he was ever going to make any investigation with respect to these drugs, because he was the lead researcher on the Human Genome Project. And that was a very sophisticated exercise, the mapping of the human genome. And I've got some intimate knowledge about that because my brother, who's an associate professor in genetic epidemiology, was a member of the Human Genome Project. And so it's been in our family for decades now. Now, Kevin McKernan finally received, well, he came into some vials of both Pfizer and Moderna. And, there's a backstory there. But he's the guy, if you want to go to anyone and say, what's in this substance? He's the guy who knows all the tests because he developed all the mapping tests for determining what a particular substance might be made up of. And so he received Pfizer Moderna vaccines and he ran them through his sequencing PCR, qPCR initially, and put out his preliminary findings in February of last year and said, whoa, everybody, hold on. Our first results are these things are not just a little contaminated with synthetic DNA, they are grossly contaminated. And words had to be used like excessively contaminated. Now, one of the measures from the early series of tests, because it took him quite a few attempts to nail down, quantify. When you've got new chemicals, which is what these are, they're regarded as a new biological entity. You have to match certain what's called primers to be able to properly locate, DNA, for instance, in these vials amongst all the LNP material, amongst all the mod RNA. In other words, separate all that stuff out and say, okay, what's left? Is that DNA and how much of that DNA is there? And so he went through a series of tests and then speaking with various colleagues. We got to figures like, just to be clear, a little backstory, mod RNA requires plasmid DNA to create that mod RNA.
From a bacteria that the RNA
That's
is
right, yes.
synthesized in. Yeah.
And when the mod RNA is created, it's all in one big vat. They're meant to put in these what's called DNases. The DNase is essentially a detergent which it's not meant to affect the mod RNA, but it'll break up all the DNA that created
from the cells that produce the RNA, the E. coli cells. That, yeah.
And then that broken down DNA is then meant to be filtered away, separated.
Yeah.
So then you've just got the mod RNA, which then they combine with the LNP. But with any manufacturing process, there's always the risk of a little bit of, let's say, residual product from the manufacturing process. And that's been known because, for decades now, that for that particular technique, creating synthetic RNA from synthetic DNA, that's been going on for decades. So just to be clear to all your viewers. And it's been recognized for decades that sometimes the final product will have a little bit,
tiny bit,
a very tiny bit of that broken up DNA, and it's called residual template or
Yeah.
residual DNA template. Now, the FDA, and it's the same guidelines that have basically been followed globally, TGA follows the same thing, has recognized for several decades that no more than 10 nanograms of residual DNA template should be present in any one particular dose. That's a very, very small, very, very small amount. What Kevin McKernan discovered was that that limit of 10 nanograms has been exceeded by as much as 509 times greater. So we're talking about literally thousands of times above the legal limit. And here's where the scary part is. That 10 nanogram limit per dose was in respect of what was recognized and always understood as naked DNA. Naked DNA. So that is to say, there's just a little fragment of DNA all by itself floating in the syringe that goes into you. Naked DNA, when it hits the bloodstream, a little fragment of something else, your immune system just recognizes. It's called exogenous. Like, oh, there's a fragment of foreign DNA. I'm just going to go and scoop that up. That's rubbish, right? We always get foreign DNA in us from just our surrounding environment, the food we eat, everything. And the body's just used to picking it up like trash, like it does pick up so many other things.
Yeah.
But in this instance, and Kevin McKernan and his crew, his lab crew, were able to go and subsequently verify this. Just like the mod RNA subsequently in the processing, manufacturing process gets enveloped, encapsulated in the LNPs, these fragments of DNA are also being protected by the LNPs as well. And so, pardon me, that's a cloaking mechanism.
Yeah.
That's a cloaking mechanism. Okay.
So the DNA actually gets surrounded in its own lipid nanoparticles, so it's fully protected.
That's right. It's fully protected
through the bloodstream. The nanoparticles will take it through the cell membranes.
You just laid it out for us, John. And so it's given the passport. It's given the free passage to buy it. Go to the testes, go to the ovaries, go to the spleen, lungs, liver, and brain. Present to the cells in any one of those organs, and then transfect into the cell. Now, let's be clear. When I use this example, you see me talking about one lipid nanoparticle complex. Now, the other science that they didn't tell you about these things is that the lipid nanoparticles, the LNPs, they tend to move as a group. It's all to do with their static charge, their electric charge. Okay. And so if the static charge on the actual cluster...
Cohesive effect, I think it's called.
That's right. It's got to do with zeta potential, these other terms.
Yeah.
But depending on one individual to the next, you can very well expect that because of this static charge, there might be a pull, like a lot of the LNPs will be pulled, let's be properly graphic here, pulled to my testes, right? There'll be a whole bunch of these LNPs, and we're talking in the billions.
Yeah.
Tens of billions or hundreds of billions. And so we'll get a whole bunch of these LNPs in the billions, perhaps in my testes, presenting to the cells there and then delivering not only the mod RNA, but also significant quantities of the synthetic DNA. Okay, this is where my spermatozoa is created. That's meant to create my offspring. Now, this DNA is being delivered as efficiently, it's called efficient transfection. Like how well do we get this stuff into the cells? Well, it gets delivered very efficiently.
Aren't we clever?
Now we spoke about the ability of the mod RNA to be reverse transcribed into DNA. It's a completely simplified matter when it comes to the DNA. Now, a lot of this DNA fragment, particularly the Pfizer, because it's got the SV40 enhancer and promoter sequence, not the entire SV40 genetic sequence, which was found to have poisoned the polio vaccines of the 1960s and created decades of cancers afterwards. But just the SV40 enhancer and promoter, that's been used by scientists for a long time because it's super, super efficient at gaining entry into the nucleus of the cell. The enhancer has got, like I mentioned with the spike protein before, it's got a nuclear localization signal, but much, much more efficient. Much more efficient.
Straight into the nucleus of the cell.
Yeah.